Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome to the Deep Dive. Today we're getting into something really cutting edge, but also, well, surprisingly fragile. We're talking about biologic therapeutics, monoclonal antibodies, therapeutic proteins. These are the drugs really changing the game in areas like cancer and autoimmune disease.
They really are revolutionary.
Yeah.
But the very thing that makes them so effective, their enormous size and complexity, that's also their biggest challenge when it comes to making them.
How so?
Well, they're massive molecules. They're produced by living cells, not simple chemical synthesis. And that brings a whole set of unique stability problems that, regular small molecule drugs just don't have.
Okay, so complexity means fragility. That sounds like the core issue.
Exactly.
So the big question for the companies making these, and really the mission for our deep dive today, is pretty fundamental, isn't it?
It is.
How do they guarantee that every single batch, every vial that reaches a patient is actually safe and effective, especially considering all the stresses these things face?
Right? Stresses during manufacturing, during shipping, even just sitting on a shelf in a hospital pharmacy.
Yeah.
So our job today is to sort of unpack this comprehensive control strategy they use. It covers the entire life cycle of the drug. It's built around something the industry calls critical quality attributes, or CQAs.
CQAs.
Yeah, we're basically tracing the science they use to ensure clinical consistency batch after batch, right from the lab bench all the way to, the drug going into a patient's vein.
Okay, let's start right there then with that fragility, because I think most people, myself included maybe, don't really grasp just how delicate these things are. Can you give us a sense of the scale difference?
Absolutely.
Yeah.
Let's take aspirin. Everyone knows aspirin, right?
Yeah.
It's tiny, maybe 21 atoms, weighs about 180 grams per mole, chemists would say.
Okay.
Now, compare that to a typical monoclonal antibody. You're looking at structures with over 1,300 amino acids.
Wow.
And they weigh around 150,000 grams per mole. It's a completely different league.
So you're saying it's like comparing, I don't know, a grain of salt to something almost the size of a tiny microbe. That scale difference is huge.
It really is. And that massive size means it has this very specific, intricate, three-dimensional shape. It's conformation. That shape is absolutely essential for it to work biologically.
Right.
But here's the catch. That structure is only marginally stable.
Marginally stable?
Yeah. It's held together by relatively weak forces, things like hydrogen bonds, hydrophobic interactions. These are easily disrupted. Think of it like a very complex, delicate piece of origami.
Which means it doesn't take much for it to fall apart, basically.
Precisely. There are several ways these molecules can degrade.
Okay, let's walk through those. What are the main physical ways they break down? Aggregation sounds like a big one you mentioned earlier.
Aggregation is definitely a primary concern, maybe the primary concern, especially because it can trigger an immune response.
Mm-hmm.
This is where the individual protein molecules start sticking to each other. They clump together, form dimers, bigger groups called oligomers, sometimes even visible particles.
And what causes that clumping?
It happens whenever that delicate native structure gets even slightly disturbed. It exposes these internal parts of the molecule, these sort of sticky hydrophobic regions that normally face inwards. Once they're exposed, they want to get away from water, so they glom onto each other.
And the triggers for that disturbance are everywhere in the process, right?
They really are. Temperature changes up or down, shifts in pH, the acidity, even just physical stress, like pumping the liquid through pipes or simple agitation.
Even shaking it?
Even shaking it. And another thing is the concentration. These drugs often need to be highly concentrated to be effective, but that high concentration itself pushes the molecules closer together, increasing the chance they'll aggregate.
Okay, aggregation is bad. What else physically? Denaturation.
Right, denaturation, that's the complete loss of that specific 3D shape. It unfolds, basically.
And if it unfolds,
It loses all biological function. It's useless. We typically think of heat causing this, thermal denaturation. Proteins unfold between, say, 40 and 80 degrees Celsius.
makes sense.
But what's really interesting, and maybe less intuitive, is cold denaturation.
Cold denaturation, things breaking down because they're too cold.
Exactly. Near freezing temperatures, around 0 Celsius.
Yeah.
It's not driven by heat energy, but by changes in how water molecules interact with the protein surface at low temperatures. It can also cause unfolding.
That's wild. You try to freeze something to keep it stable, and the cold itself can damage it.
It's a real challenge. And then there's another physical issue, adsorption.
Adsorption, like sticking to surfaces.
Precisely. We talk about big things like temperature, but simple contact with surfaces is a constant issue.
Is it really a major factor?
Oh, absolutely. Proteins are inherently sticky, especially to surfaces.
Right.
Glass vials, plastic tubing, the filters used in purification, they'll stick to all of it. And critically, they adsorb to the air-liquid interface, the surface of the liquid in a partially filled container. Shaking or swirling increases that surface area dramatically.
And when they stick?
That sticking can slightly change their conformation, their shape. And that slight change can be the trigger that starts the whole aggregation cascade we just talked about. It leads to clumping, and you lose product.
So physical breakdown is a minefield. What about chemical changes?
Chemical degradation is another major route. Oxidation's a really common one.
Like resting, but for proteins?
Sort of, yeah. It's
Hmm.
often triggered by exposure to light or even tiny trace amounts of metal ions left over from the process. It mainly attacks specific amino acids like methionine and tryptophan.
And when those get oxidized?
The protein structure changes. That can induce aggregation, just like physical stress. And often it directly destroys the part of the molecule responsible for its biological activity.
So another way to lose function, and you mentioned one more.
Deamidation, yes. This is a chemical modification that happens mainly to an amino acid called asparagine. It converts this neutral residue into a negatively charged one, aspartic acid.
So the overall electrical charge of the protein changes.
Exactly. You end up with what are called acidic charge variants. The drug molecule now has a different electrical profile.
Okay, that's a lot of ways things can go wrong.
And what's really critical to understand is that these pathways often interact. They're interconnected.
How so?
Well, a small temperature fluctuation might cause a little bit of unfolding partial denaturation. That exposes some of those internal amino acids, making them vulnerable to oxidation. And at the same time, the slightly unfolded molecule is now stickier, more prone to aggregation. It's like a domino effect.
Wow. Okay, so given this inherent instability, this multi-pronged fragility, just testing the final product at the end clearly isn't enough. You can't just hope for the best.
Absolutely not. That's where this whole proactive approach comes in, known as quality by design, or QBD.
Quality by design.
Right. And the absolute core concept within QBD is the critical quality attribute, the CQA we mentioned earlier.
Okay, define that for us again. What exactly is a CQA?
So the official guidelines, like from ICH, define a CQA as any physical, chemical, or biological property or characteristic.
Okay.
That needs to be kept within a very specific, appropriate limit, range, or distribution to ensure the desired product quality.
So it's the measurable stuff that actually matters for the drug working safely and effectively.
Precisely. CQAs are the scientific bridge connecting the drug's properties, how it's manufactured, and ultimately how it performs in the patient.
So QBD then, it sounds like the industry shifting from, inspecting quality after the fact to meticulously planning and building quality into the entire process from the very beginning.
That's a great way to put it. It starts way upstream. You first define exactly what the ideal final drug product should look like and do. That's called the Quality Target Product Profile, or QTPP.
The target profile.
Right. Then you work backwards, using risk assessment focused on the patient, to identify which specific attributes of the molecule and the process are truly critical to achieving that target profile safely and effectively. Those become your CQAs.
And how do they actually control these CQAs during manufacturing? The guidelines mention some key tools, right?
Yes, ICH-Q11 highlights a couple of key enablers. First, there's Process Analytical Technology, or PAT.
PUT. That involves using sensors during the process itself, doesn't it? Like monitoring things in real time.
Exactly. Instead of taking samples and waiting hours or days for lab results, PT uses inline or at-line sensors. These sensors continuously monitor critical parameters and attributes during the manufacturing process.
So quality is being checked and verified as it's being built.
Built in, not tested in, is the mantra. If a parameter, say pH or temperature, starts to drift slightly outside the ideal range, the system catches it instantly, allowing for immediate correction.
That real-time data and understanding must feed into the second key concept you mentioned, the design space.
It absolutely does. The design space is essentially the scientifically established operating range for the process. It's a multi-dimensional combination of process parameters and material attributes like temperature ranges, pH ranges, flow rates, raw material specs that has been demonstrated to consistently produce a product meeting its CQAs.
So it's like a pre-approved operational playbook for the process.
Exactly. And if manufacturers can prove to regulators they deeply understand their process and can reliably operate within this validated design space.
They get more flexibility.
Huge flexibility. It means they can often make small improvements or adjustments within that defined space, maybe optimize yield slightly or adjust for a new raw material lot without needing to go back to the regulators for approval every single time. That saves enormous amounts of time and money.
Okay, so to define and monitor that design space and control those CQAs, you need the right measurement tools. What's in the analytical toolkit for biologics?
It's quite sophisticated. For measuring those size variants, especially the aggregates we worry about, the workhorse technique is size exclusion chromatography, or SEC.
SEC. How does that work?
Think of it like a molecular sieve. You pass the protein solution through a column packed with porous beads. Larger molecules, like aggregates, can't enter the pores easily, so they pass through the column faster. Smaller, correctly sized molecules take a more tortuous path through the pores and come out later.
So it separates based purely on size.
Correct, allowing you to quantify the amount of aggregates versus the desired monomer.
Okay. And what about those charge variants, the acidic ones from deamidation or other types?
For charge variants, we primarily use ion exchange chromatography, IEX, or a technique called capillary isoelectric focusing, CIEF.
Ion exchange based on electrical charge.
Exactly. IEX uses a charged column matrix. Proteins with different overall charges will bind to the column with different strengths, allowing them to be separated and quantified as they are eluted off the column. CIEF separates them based on their isoelectric point in a capillary.
Got it. And confirming the basic structure, making sure the amino acid sequence is right, and identifying chemical changes like oxidation.
For that level of detail, the gold standard is peptide mapping, usually coupled with liquid chromatography, mass spectrometry, or LCMS.
Peptide mapping? Sounds complex.
It is quite involved. You basically use enzymes to chop the large protein into smaller, predictable peptide fragments. Then you separate these peptides using LC and feed them into a mass spectrometer. The mass spec measures the precise mass of each peptide fragment.
And that tells you.
By comparing the measured masses to the expected masses based on the known amino acid sequence, you can confirm the sequence is correct, and you can pinpoint any modifications like oxidation or deamidation because they change the mass of specific peptides in a predictable way.
Okay, that's a powerful set of tools for control. Now let's connect these molecular details back to the patient. Why is aggregation such a clinical concern? You mentioned immunogenicity.
Right. It's about more than just losing some active drug. Aggregation is probably the single biggest factor driving the risk of immunogenicity.
The body reacting against the drug.
Exactly. Because aggregates are often particulate, and they have these repeating structures or epitopes on their surface. They look very much like a virus or a foreign invader to the immune system. They're really potent triggers. They get readily picked up by antigen-presenting cells, which then kickstarts an immune response, leading to the production of anti-drug antibodies, or ADAs.
And ADAs are bad because...
Well, at a minimum, they can bind to the drug and neutralize it. The therapy simply stops working, which is obviously a disaster for the patient relying on it.
That's bad enough.
But in some severe cases, it gets worse. The ADAs generated against the drug might cross-react with the patient's own natural version of that protein if one exists.
Oh, wow. Like an autoimmune reaction triggered by the drug?
Precisely. The classic tragic example is the Eprex case from years ago. Eprex is a recombinant human erythropoietin, a drug that stimulates red blood cell production. A change in its formulation was later linked to a sudden spike in cases of a condition called pure red cell aplasia, or PRCA, a severe type of anemia.
What happened?
It turned out patients were developing neutralizing ADAs against the Eprex. But crucially, these ADAs also targeted the patient's own natural erythropoietin. The immune system essentially shut down the body's ability to make red blood cells. Aggregation in the new formulation was strongly implicated as a contributing factor to that increased immunogenicity.
That's a terrifying example of the stakes involved. Okay, let's shift to another CQA, glycosylation. The sugar chains attached to the protein. You said this one is interesting because variability isn't always bad.
That's right. Glycosylation is fascinating because unlike the protein sequence itself, which is dictated by the gene, the pattern of sugars attached is determined by the manufacturing process, the cell line, the bioreactor conditions. It's highly process dependent.
So it can vary.
It can. But here's the key. Sometimes specific types of variability can actually be engineered to be beneficial.
How so? Beneficial for efficacy.
Yes, particularly in cancer therapies. Take certain monoclonal antibodies designed to kill tumor cells. There's a specific sugar called fucose.
Yeah.
If you engineer the manufacturing process to produce antibodies that lack this core fucose sugar, a process called afucosylation.
Afucosylation, okay.
These afucosylated antibodies bind much more strongly to certain receptors on immune cells, specifically FCC gamma RIIA on natural killer cells.
Stronger binding means.
It means significantly boosted ADCC antibody-dependent cell-mediated cytotoxicity. Basically, the antibody becomes much, much better at directing the immune system to kill the target cancer cell.
So you can deliberately tweak the sugar pattern to make the drug more potent. That's incredible.
It is. And glycosylation also impacts pharmacokinetics, how long the drug stays in the body.
How does that work?
Certain sugar residues, particularly sialic acid, can sort of mask the antibody from clearance mechanisms in the liver. The more sialic acid residues on the sugar chains, higher sialasia, the longer the drug tends to circulate in the bloodstream. Its half-life increases.
Longer half-life is generally good, right? Less frequent injections for the patient?
Usually, yes. It can significantly improve convenience and adherence.
And you mentioned charge variants earlier arising from things like deamidation. Do they affect clinical performance too?
They can, yes. Those acidic variants created by deamidation, if they occur in or near the part of the antibody that binds the target, the CDRs, they can sometimes reduce the drug's potency.
Okay, so acidic variants may be less potent. What about other types?
Interestingly, some basic variants have shown effects on pharmacokinetics. These can arise from, for example, incomplete processing of the antibody's C-terminus, leaving an extra positively charged lysine residue.
A basic variant, okay.
There's evidence that these basic variants can bind more strongly to a receptor called the neonatal FC receptor, or FcRn.
FcRn, what does that do?
FcRn is like a recycling system for antibodies. It grabs antibodies inside cells and protects them from being broken down, then releases them back into circulation.
Ah, so stronger binding to FcRn means?
It means the antibody gets recycled more efficiently, sticks around longer. So, like increased sialation, certain basic charge variants can also translate directly into a longer serum half-life for the drug.
Again, potentially meaning less frequent dosing for patients.
Exactly, which really underlines the point we touched on earlier.
That the goal isn't zero variability.
Right. The goal of the control strategy, driven by QBD, isn't necessarily to eliminate all variability. Some is inherent when you're using living cells. The goal is to deeply understand it, to manage it within validated limits, and in some cases, even leverage specific variations to improve the drug's clinical profile.
That makes perfect sense. Okay, so the drug is made, it's characterized, it meets its CQA specs, but the journey isn't over. We hit what you called the last mile challenge, getting it safely to the patient.
Correct. And the first line of defense here is the formulation itself. The liquid the protein is dissolved in.
What's in the formulation?
It's carefully designed. It contains buffers to control pH, salts to control ionic strength, and often stabilizing excipients like sugars, sucrose, trehalose, or surfactants like polysorbate 80. These create a protective little microenvironment for the protein in the vial.
But then it gets to the clinic, the hospital pharmacy, the bedside, and that introduces a whole new set of risks, right? Especially during preparation for infusion.
Absolutely. This is where sterile compounding and IV admixture happen, usually governed by strict standards like USP 797 and 800, but it's a high-risk phase. A key issue is that the drug, which is often highly concentrated in its protective formulation, usually needs to be diluted significantly before it can be given as an IV infusion.
And diluting it reduces the concentration of those protective ingredients.
Exactly. The protective effect of the stabilizers gets weakened. But the even bigger, more acute risk is diluent incompatibility.
Incompatibility.
Yes. Remember, we said many antibodies are formulated at a slightly acidic pH for optimal stability in the vial. Now, imagine a nurse or pharmacist takes this concentrated acidic antibody solution and mistakenly dilutes it in D5W dextrose 5% in water. D5W is essentially unbuffered sugar water.
What happens?
The pH of the diluted solution will rise significantly, potentially approaching the antibody's isoelectric point, the pH where its net charge is 0. At this point, proteins are least soluble and most prone to aggregation and precipitation. You can get rapid, massive aggregation, even visible cloudiness, right there in the IV bag or syringe.
That's horrifying. You'd be creating aggregates right before infusing them into the patient.
It's a major recognized clinical risk. And it's precisely why, for the vast majority of monoclonal antibodies, the required diluent specified in the instructions is normal saline 0.9% sodium chloride.
Why saline?
Because saline is buffered and has sufficient ionic strength to help maintain the protein's solubility and prevent that catastrophic isoelectric precipitation, even after dilution. The instructions for use are absolutely critical here.
Okay, so handling in the clinic is crucial. What about getting it there? The logistics. You mentioned the cold chain.
Yes. The cold chain is non-negotiable for most biologics. They typically need to be kept refrigerated between 2 and 8 degrees Celsius from the moment they leave the manufacturing plant until they're about to be used.
Keeping things cold during shipping?
It's much more sophisticated than just ice packs now. We talk about maintaining a chain of data integrity. Modern biologic shipping uses validated cold chain shippers packed with phase change materials and equipped with electronic data loggers, sometimes even GPS and IoT sensors.
So you have a continuous record.
A continuous auditable record of the temperature the product experienced throughout its entire journey. This data proves the cold chain wasn't broken. But even with all that tech, the last mile, getting the drug from the regional distribution hub to the actual clinic, the hospital, or sometimes even a patient's home, that's widely considered the most vulnerable phase.
Why is that?
Just more handling variability, less standardized procedures perhaps, more potential exposure to uncontrolled ambient temperatures or delays. It requires constant vigilance.
Okay, so pulling it all together, this life cycle-wide control strategy we've been discussing sounds incredibly comprehensive. It requires tight integration across so many different areas.
It really does. You need deep expertise and seamless collaboration between formulation development, process development and manufacturing, analytical sciences for testing and characterization, supply chain and logistics, and finally, clinical pharmacy practice to ensure it's handled and administered correctly. Clear instructions for use are paramount.
It's a whole ecosystem of control.
And ultimately, all of this rigorous science, this deep understanding generated through the QBD approach, builds the compelling narrative that regulators need to see. The goal, again, isn't claiming zero variability that's unrealistic for biologics made in living systems. The goal is demonstrating profound understanding of the sources of variability and proving you can consistently control that variability within a predefined, clinically justified design space.
That robust, data-rich story is what gets the drug approved and keeps it safely on the market. Which brings us to maybe a final provocative thought for you, our listeners, to chew on. We've established that controlling variability is crucial to prevent bad outcomes, like immunogenicity.
Right.
But we also touched on how engineering specific types of variability, like a fucosylation for better cancer killing or higher salylation or basic variants for longer half-life, can actually create better drugs.
Yes, deliberately introducing and controlling specific modifications.
So if manufacturers get really good at creating and, crucially, controlling these beneficial variant profiles, making drugs that are intentionally different, maybe even superior to the original reference molecule, what does that mean for the future?
It raises fascinating questions, doesn't it?
Yeah. How will regulators adapt? How do you define the quality standards and acceptance criteria for a drug that is designed to be different, to be intentionally variant in a beneficial way? Where do you draw the line?
It's a complex challenge for the next generation of therapies and the agencies overseeing them. How do you manage specifications for intentionally engineered variability?
Definitely something to think about. It really highlights how dynamic this field is. That seems like a perfect place to leave it for this deep dive. Thank you so much for walking us through that complex world.
My pleasure. It was great discussing it.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.